bacteria have these things called periplasmic binding proteins. This is Leninger Biochemistry 2nd edition. So these are cool. Bacterial cells, think of them like submarines. They have two hulls. They've got the pressure hull, where the people live, and where there's air, and where it's a healthy space, right? That's where the engine is. That's where stuff is happening, right? And then you have the outer hull, and then in between it you have sort of this dead space that holds the extra water. That's the ballast, so you can blow it out, and it's where you put extra storage for shit that you don't really need. It's just it's sort of like an attic space. Okay, so in the attic, these bacteria keep these periplasmic binding proteins, and they drop them, they throw them out, they blow them out into the water, so to speak. And all these binding proteins, each one, there's a bunch of them, and each kind of them, right? A bunch of kinds of them. And they each bind to, some of them bind to amino acids, some of them bind to sugars, some of them bind to other ions. So they're each going after one specific kind of thing, right? And then they think, here, specific periplasmic binding proteins have been isolated for most of the common amino acids, and for glucose, galactose, arabinose, ribose, and other sugars. Those are all sugars. All the ose are sugars. They have molecular blah, 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 blah. Some of them do stuff to ligands, blah, blah, blah. And they think that that's how bacteria know what to eat. Yeah. So they blow these out, and then it attaches onto this stuff, and then the bacteria uses that to know what they need to be eating, so that when they bump into it, they're like, oh, this is one of my tags for food. This is food. Ah, yes, okay, food. Come in. No. That's pretty cool. Evidence has suggested that the periplasmic binding proteins are dissociable components of membrane-bound transport systems that contain the specific substrate recognition sites for their substrates. That's what I just said. They are also believed to be receptor sites in chemotaxis, by which bacteria recognize nutrients and propel themselves by flagellar motion in the direction of increasing nutrient concentration. Okay, so then, so you're like, oh, I know there's food around here somewhere, but I can't see because I'm a bacteria, and bacteria don't have eyes. So, I just dump out the periplasmic binding proteins, and then I look around and see if I caught anything. Are any of them attaching to food? Is there food? Where's their food? Is that? Nope, nothing there. Yeah, this has food on it. This is attached to food. Hey, more food over here. Hey. It also means that if you have a bunch of bacteria of the same species, they're all going to be dumping the same kind of periplasmic binding proteins, right? So the more of them that are there, the more of these periplasmic binding proteins will be present, and therefore the better they'll work, because all the available food particles will be saturated with these periplasmic binding proteins, so that you'll be constantly bumping into them as bacteria because everybody's making them. That's very clever. Let's play some piano.